Okay, once again, thank you so much for your time here. Yeah, it helps me remember these processes by remembering where they happen, but replication in the nucleus, trying to copy all of the DNA, transcription, just trying to copy or replicate a small piece of the DNA, really you make it into an RNA so it can float around in the nucleus. Translation is the same thing as protein synthesis. Okay, where do they happen? Replication and transcription. You need DNA for those reactions to happen. That's going to happen in the nucleus. Translation, really anywhere. Anywhere out here in the cell. Anywhere a protein is needed. Okay, so there's another picture. Replication, transcription, nucleus, translation out in the cytoplasm. All right, please, we drew these in class. If you can draw the 1, 2, 3, 4, if you can draw the 5-sided sugar and understand our convention for numbering those carbon atoms, then you can really understand all of our ways we model the genes and model our DNA. Yes, please know the nitrogen base is attached to carbon 1. Carbon 2 is how you can tell what type of sugar it is, whether it's ribose or deoxyribose. And then really at 3 and 5, that's where the phosphate ions will attach. Okay. So yes, if you missed those drawings in class, here they are available for you, but let me know if you need a better copy. There's more links on the website below of these same drawings. So yeah, when we draw the whole thing, I guess just kind of try to remember that hydrogen bonds hold the nitrogen bases together, phosphodiester bonds maintain the integrity of that backbone or hold the sugars and phosphates together. You can also see from this drawing how we label one side 5 to 3, 5 to 3, 5 to 3. The other side is complementary or anti-parallel, and it's labeled 3 to 5, 3 to 5. And once again, that's just from counting the carbons and the way we number them in the sugar molecule. All right, hopefully you've studied this slide. This is from our National Institutes of Health. Really the same pictures, I believe, kind of show up in the Nature article. But yeah, just different ways to represent DNA and RNA. You can see the structure of all the nitrogen bases there. And once again, keep in mind the hydrogen bonds hold those nitrogen bases together in the middle. You can break those hydrogen bonds and really not fundamentally change what the DNA is. Just changing how those nitrogen bases are stuck together, but they'll still keep the same sequence. The sugars and phosphates will still stay bonded together. Hydrogen bond is not a covalent bond. The phosphodiester bond is a covalent bond, though. All right, so let's take these models that you've seen, hopefully before this class, and look at maybe this is something you've never seen. In a lot of ways, we're very lazy with this. So they're just saying, okay, piece of DNA, there it is, piece of DNA. Let's start labeling some pieces of it. They label some pieces of it with letters. They label some pieces of it with numbers. Altogether, this little thing here, this gene, has 7,700 nitrogen bases in there. Actually, there's the base pairs. So yeah, it's double-stranded DNA. So here's a little piece with 7,700 base pairs. And this is a gene that makes the albumin that shows up in an egg. Okay, albumin's a protein. All right, this DNA, the enzymes come in. The enzymes allow the transcription to happen. So here's the piece of RNA. Keep the same letters, keep the same numbers there. I guess we can kind of assume it probably is close to the same number of base pairs. Then all of a sudden, all these yellows, the A's, the B, C, D, E, F, G, those are gone, cut out. They don't show up down here. So we went from 7,700 nitrogen bases to 1,872 nucleotides, which one nucleotide will have